Let's dive right into this explainer. Have you ever actually thought about the microscopic machines that quite literally make human life possible? I mean, every single second, millions of incredibly complex chemical reactions are just firing off inside your body. But how does your system manage to run all these intense processes without totally breaking down? Well, today, we're going to unlock the biological cheat codes that make it all happen. Enzymes. So let's start with a massive biological problem. Chemical reactions require a huge amount of energy and heat to sustain life naturally. Just think about the sheer thermal energy you need to ignite a log or to get a pot of water to boil. If our cells had to generate that kind of intense heat just to spark the reactions needed to digest a sandwich or, copy our DNA, our cells would be instantly destroyed. We would literally boil over from the inside.
But obviously, we don't boil over. And the elegant solution to this biological heat problem is the enzyme. An enzyme is a highly specialized protein that acts as a biocatalyst to efficiently speed up these reactions. And I mean speed up. They can enhance the rate of cellular reactions up to 1,000,000-fold, 1,000,000 times faster. And the truly amazing part here is that they aren't consumed in the process. Once the reaction is done, the enzyme just releases, totally unchanged, and is immediately ready to go to work on the exact next molecule.
Today, we're going to zoom all the way from the micro-level physics of activation energy right out to the macro level of human anatomy. Here's our roadmap. 1. The spark of life. 2. Lowering the energy barrier. 3. Locks, keys, and handshakes. 4. When enzymes break down. And finally, 5. Keeping you alive.
All right, moving into section 2, lowering the energy barrier. Let's get into the actual physics of enzymes. Transition state theory perfectly illustrates how this works. Think of a chemical reaction like climbing a massive mountain. Without an enzyme, that peak, the uncatalyzed reaction, is a huge, exhausting climb. But when you introduce an enzyme, that steep energy barrier is drastically reduced. The start and end points of the reaction, the overall energy change, stay exactly the same, but that massive hurdle right in the middle practically disappears. So in plain English, enzymes create an incredible biological shortcut. They lower the activation energy, which allows these vital, life-saving reactions to happen rapidly at a perfectly normal body temperature. By stabilizing that middle transition phase, the enzyme effectively removes the need for cellular fire. It lets you happily process energy at a mild 98.6 degrees Fahrenheit. Okay, section 3, locks, keys, and handshakes. Let's talk about how enzymes actually bind to things. Way back in 1894, a chemist named Emil Fischer proposed this incredibly specific lock and key model. He suggested the enzyme's active site is a strictly rigid lock, and the substrate, which is just the target molecule, is the perfect, unchangeable key. It's actually a great way to visualize just how highly specific enzymes are. I mean, they don't just grab anything passing by, they only bind to their one perfect match. But, nature is rarely that perfectly rigid. And that brings us to Daniel Koshland's 1958 induced fit model. Where the old lock and key model imagines a stiff, inflexible lock, the induced fit model proves that the enzyme is actually dynamic. It physically molds itself around the substrate the second they make contact. Think of the induced fit model less like shoving a key into a metal lock and more like a warm, firm handshake. The molecule approaches and the enzyme undergoes a conformational change. It literally wraps around it. This dynamic squeezing action puts severe stress on the substrate's bonds. It physically forces it into that stabilized transition state we talked about earlier, and boom, that is exactly what triggers the catalysis to occur. Which brings us to section 4, when enzymes break down. Let's explore the limits of life. Because enzymes are so specialized, they have a very strict Goldilocks zone. They need conditions to be just right to function properly. We're talking specific temperatures, very strict pH levels, usually right in the physiological range of 6 to 8, and they even sometimes need helper molecules called cofactors to get the job done. If the environment strays outside of those super precise parameters, the enzyme simply can't perform that perfect handshake. So, keeping that in mind, have you ever wondered why high fevers are so incredibly dangerous to the human body on a molecular level? Or why sudden, extreme changes in blood acidity can be lethal? Well, when your temperature spikes too high or acidity drops too low, the enzyme goes through something called denaturation. This means it permanently loses its shape. The protein literally unfolds and warps. The lock is completely, irrevocably broken. And when that active site shape is destroyed, the target molecule can't bind anymore, which means those life-sustaining cellular reactions just stop. They grind to a sudden, complete halt. Moving on to Section 5, Keeping You Alive. Let's look at how enzymes are working in your body right now. To put this in perspective, you have lipase and trypsin working tirelessly in your gut and small intestine right this very second, actively digesting your last meal into usable fuel. But it's not just human digestion. Up in your cellular nuclei, an enzyme called helicase is actively unzipping your tightly wound DNA coils. It's clearing the path for another enzyme, DNA polymerase, to synthesize brand new strands so your cells can replicate. Without these biocatalysts, cell division would be absolutely impossible.
And we aren't just relying on the enzymes we were born with anymore. Science is using them as incredibly powerful therapeutic tools. We track them in blood panels to diagnose organ failure. We actually administer them as enzyme replacement therapies for severe genetic storage diseases like Gaucher's or Pompe's disease. We even use specialized anti-tumor enzymes to systematically starve cancer cells of the specific amino acids they need to survive. It is truly mind-blowing stuff.
I want to leave you with this final provocative thought. Way back in 1964, scientists paved the way for creating completely synthetic enzymes in the lab. As we mastered the ability to artificially assemble these custom-built catalysts, which we call synzymes, from scratch, what incurable diseases might we conquer next? Could we design perfect, tiny machines to clean up specific toxins in our blood or even reverse cellular aging? The possibilities are quite literally endless. Thanks for joining me on this explainer today.